Dr. Mitch Robinson, and this video is on glucose metabolism. It accompanies handout number 5 for cellular and molecular medicine. In this video, we will examine the cellular metabolism of glucose and its conversion to pyruvate through glycolysis. We've looked at the structure of carbohydrates and their digestion and the transport of monosaccharides into cells, and now let's look at the pathway for the conversion of glucose to pyruvate through glycolysis. I'm sure most everyone's familiar with the pathway for glycolysis, and we're not going to take too much time with the individual reactions and enzymes, but we do need to look at some aspects of glycolysis that are important in metabolism, and particularly in metabolic diseases. For now, let's examine the pathway for glucose metabolism, and later we'll see how other monosaccharides, fructose and galactose, enter the pathway. And we'll look at the complementary pathway, gluconeogenesis, where pyruvate is converted back to glucose. Glycolysis is the metabolic pathway that converts glucose into pyruvate, and the free energy released is used to form high-energy compounds ATP and NADH. Glycolysis occurs in all tissues of the body and nearly all organisms, both aerobic and anaerobic. Glycolysis plays a dual role in catabolism, serving as a means of anaerobic energy production, or fermentation, and as a means of generating the precursor molecules for aerobic energy production, respiration. In fermentation, the final electron acceptor will be an organic molecule. This can involve transfer of the electrons back to pyruvate to form lactate, and this is known as homolactic fermentation. Glycolysis also serves as an entry to aerobic metabolism. In this case, molecular oxygen serves as the final electron acceptor, and this is known as respiration. Let's look at glycolysis in the big picture of metabolism, shown on page 3. We'll come back to this map of metabolic pathways often to see how each pathway fits into the big picture. We begin here with glucose and the steps in glycolysis leading to the production of 2 molecules of pyruvate. Pyruvate can be converted to lactate anaerobically, or it can enter the mitochondria where it is metabolized aerobically to CO2 through the TCA cycle. And again, oxygen is the final acceptor of electrons. When glucose enters the cell, it is immediately converted to glucose 6-phosphate. The enzyme that converts glucose to glucose 6-phosphate is hexokinase. There are 4 mammalian isoenzymes of hexokinase. These are known as type 1 through type 4. Type 4 hexokinase is known as glucokinase. To simplify things, we usually refer to type 1, 2, and 3 as hexokinase and type 4 as glucokinase. The table on page 4 shows the location and properties of glucokinase and hexokinase. Glucokinase is found in liver, kidney, and the B cells of the pancreas, while hexokinase is found in most other tissues. Glucokinase is specific for glucose, while hexokinase phosphorylates other monosaccharides, including fructose and galactose. Glucokinase has a much higher Km for glucose than hexokinase. The Km is around 10 millimolar, while that of hexokinase is around 0.1 millimolar. Remember that GLUT2 is present in the liver, and it's the high-capacity bidirectional transporter. So this means that the level of glucose in hepatocytes is pretty much in equilibrium with the level of glucose in the blood. As shown in page 4 on the figure, glucokinase is operating with a Km near the level of blood glucose. What that means is it's more active when blood glucose is high, and it's less active when blood glucose is low. So this allows glucose to be actively phosphorylated whenever the level of glucose in the blood and in the liver is high, and it slows phosphorylation when the level of glucose in the blood is low. And this is important because in the liver, glucose needs to be released into the bloodstream. Hexokinase, on the other hand, has a low Km. That means that it's operating almost always at a maximum velocity. And that's important because we need to move glucose into the brain even when the level of blood glucose in the blood is low. Hexokinase is inhibited by the product of the reaction, glucose 6-phosphate. So when the use of glucose in the cell slows, glucose 6-phosphate builds up, and that inhibits further phosphorylation of glucose. So this combination of GLUT2 glucose transporter and glucokinase allows glucose to move in and out of the tissue of the liver. This combination also occurs in the B cells of the pancreas. In this case, it allows the B cells to act as a sensor for the level of glucose in the blood and to regulate the release of insulin. Now, glucose 6-phosphate is an important branch point in intermediary metabolism. And let's look at the possible ways in which glucose 6-phosphate can be metabolized. If needed for energy production, glucose 6-phosphate can be metabolized to pyruvate through glycolysis. It can also be converted to 5-carbon pentose phosphates through the hexose monophosphate pathway. Or it can be converted to glycogen if it's necessary to store glucose in this form. Okay, let's look briefly at the reactions in glycolysis so that we can identify a few points that are important about this pathway. Following the phosphorylation of glucose to form glucose 6-phosphate, there's an isomerization to fructose 6-phosphate. This creates a hydroxyl group on the 1 carbon that can be phosphorylated in the next step to form the product fructose 1,6-bisphosphate. Next, the molecule is split by the aldolase enzyme to 2 triose phosphates, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. An isomerase can interconvert these 2 molecules, so we have 2 molecules of glyceraldehyde 3-phosphate formed. The next step is an important reaction in that it uses the energy of this aldehyde of glyceraldehyde 3-phosphate to drive a reaction in which a phosphate is added. The product is this is glyceraldehyde 3-phosphate dehydrogenase, and this is a step where electrons or hydrogens are removed. 2 hydrogens are transferred to NAD to make NADH. In the end, a high-energy phosphate bond is formed. The free energy of hydrolysis of this high-energy phosphate is similar to that of ATP, and so this phosphate can be transferred to ADP to make ATP. The product 3-phosphoglycerate does not have a high-energy phosphate, but in the following steps, this phosphate can be converted to a high-energy phosphate in the molecule phosphoenol pyruvate. So, since it's a very high-energy phosphate, it can donate that phosphate to ADP to form ATP in the pyruvate kinase reaction. And the final product is pyruvate. I'll point out here that the intermediate in glycolysis 1,3-bisphosphoglycerate can be converted to 2,3-bisphosphoglycerate. And you'll recall that this is important in the regulation of oxygen binding to the hemoglobin in the erythrocyte. So you'll find a lot of this mutase in erythrocytes. Now, pyruvate, which again is the end product of glycolysis, is another important branch point in metabolism. As we mentioned earlier, pyruvate can be metabolized aerobically to CO2, or it can be converted anaerobically to lactate. Now, the enzyme for the conversion of pyruvate to lactate is lactate dehydrogenase. This enzyme uses the NADH that was produced in the glyceraldehyde 3-phosphate dehydrogenase reaction. So the conversion of glucose to lactate can be done with a balance of oxidized and reduced NADH and NAD. So there's no net oxidation reduction. So lactate is produced in tissues where there is insufficient oxygen supply to metabolize glucose to carbon dioxide. Heavy exercise, of course, causes lactate buildup in muscle tissue. Lactate also accumulates in tissue where there's a reduced blood flow or ischemia. Anaerobic production of lactate is the only way that erythrocytes can produce ATP, since they lack mitochondria. Lactic acidosis occurs when there's an accumulation of lactate that overwhelms the body's buffering ability. Humans have 2 genes for lactate dehydrogenase, or LDH, and produce 2 isoforms. The H isoform is prominent in heart muscle. The M isoform is prominent in skeletal muscle. And the LDH exists as a tetramer of 4 different subunits, either one of the 2 different isoforms. So you can have 5 different combinations of the H and L subunits to make the tetramer, as shown in this diagram on page 7. Different tissues have characteristic isoform distributions. LDH is released from cells when there's tissue damage, and the identification of the isoforms in serum by electrophoresis is a tool that will help to identify if there is tissue damage and the source of the damage. For example, elevation of the H4 form in serum indicates the presence of myocardial infarction, since heart tissue damage releases mostly the M4 form of lactate dehydrogenase. Now, it's not important that you know all of the enzymes and intermediates in glycolysis, but I do want to point out a few important features of the pathway. First, it's important to note the movement of electrons and the oxidation-reduction pathway in metabolic reactions. Note that glyceraldehyde 3-phosphate dehydrogenase is the reaction where electrons are transferred to NAD to produce NADH. And there must be a balance of the production of NAD and NADH. So these electrons will either be lost at the lactate dehydrogenase step or sent to the mitochondria for oxidative phosphorylation. Let's look at the production and use of ATP in glycolysis. 2 steps in the first part of the pathway where ATP is used, and that is glucokinase hexokinase and phosphofructokinase. And there are 2 steps in the latter part of the pathway where ATP is generated, and that is phosphoglycerate kinase and pyruvate kinase. Of course, the last 2 reactions have 2 molecules each, so we get 4 ATPs generated, and we use 2 ATPs, and so there's a net 2 ATP production. And this ATP is produced through substrate-level phosphorylation. Substrate-level phosphorylation means that we're producing ATP by directly coupling the conversion of ADP to ATP through another reaction. And later we'll look at another way to make ATP, and that's called oxidative phosphorylation. Now, it's also important to note the irreversible steps in glycolysis. There are 3 irreversible steps. The first, step 1, glucokinase hexokinase, 3, phosphofructokinase, and step 10, pyruvate kinase. These reactions all have a large negative delta G0, so they only proceed in the forward direction. These irreversible reactions are usually the steps where enzymes are regulated. Now these are the control points in glycolysis. Now glucokinase hexokinase, we've talked about the regulation of that enzyme, but it's not the main control point for glycolysis because glucose 6-phosphate, the product, can go in other directions and other pathways. So we need to regulate glycolysis at a place where the pathway is committed. And the first committed step to glycolysis that's irreversible is phosphofructokinase. So PFK is the main control point in glycolysis. PFK is active as a tetramer with 4 identical subunits. And each subunit has a catalytic site that binds the substrates ATP and fructose 6-phosphate. But it also has an allosteric site, and allosteric sites bind to molecules and regulate the enzyme. The allosteric site of PFK binds ATP as well, but binding of ATP to the allosteric site inhibits the enzyme. ATP inhibits PFK by increasing negative cooperativity for substrate. That is, the binding of one molecule of fructose 6-phosphate inhibits the binding of more molecules of substrate. And this makes for a sigmoidal velocity curve, as shown here. Now, AMP and ADP will also bind at the allosteric site, but they don't produce inhibition. So they activate the enzyme because they bind and displace the inhibitor ATP. Citrate is like ATP. It's an allosteric inhibitor. So remember that citrate and ATP bind to the allosteric site and inhibit. AMP and ADP bind to the allosteric site and activate by relieving the inhibition of ATP or citrate. AMP is probably the most important regulator of phosphofructokinase, and the question is why AMP? In most reactions where ATP is used, it's converted to ADP. But it turns out there is an enzyme, adenylate kinase, and this enzyme can take two molecules of ADP and convert them to ATP and AMP. So what happens when ATP is used is ADP accumulates, but the ADP is converted to ATP and AMP by adenylate kinase. So the net reaction is that when ATP is used, AMP accumulates, and that feeds back and inhibits phosphofructokinase. Pyruvate kinase is the third irreversible step in glycolysis. There's an isoform of pyruvate kinase in fetal tissue that has a much higher activity than in adult tissue. In adult, the slower enzyme results in some accumulation of 1,3-bisphosphoglycerate, and some of this is converted to 2,3-BPG as shown on page 6. This means that adults have more 2,3-BPG than a fetus. So, since 2,3-BPG inhibits oxygen binding to hemoglobin, in fetal erythrocytes there's a higher affinity of oxygen than in adult erythrocytes, and therefore oxygen is effectively transferred from a woman's erythrocytes to her fetus. Now, the rate of glycolysis is often very high in cancer cells. This high rate results in a high degree of anaerobic lactate production. This is thought to occur at least in part because cancer cells often express the fetal isoform of pyruvate kinase, which is much more active. The high rate of anaerobic energy production is thought to give cancer cells an advantage in tumors where blood supply and oxygen is low. Also, this does give an advantage for diagnostic imaging, which takes advantage of the high rate of glycolysis in tumors. Since cancer cells take up more glucose than other tissues, we can use radiolabeled glucose and positron emission tomography to locate tumor cells in the body. Okay, so that's the end of the video for handout number 5 on glucose metabolism.